So my name is Jacques Cohen. I'm one of the moderators today. I'm here with Professor Adashi. Both of us have interest in mitochondria, and we're welcoming you to this session regarding mitochondriacs. And of course, it's about mitochondria and reproduction. I'm honored and excited to be co-hosting this session of the International IVF Initiative. For videos of previous sessions, please go to ivfmeeting.com. Now, thank you very much for logging in. I encourage you to ask your questions using the Q&A section on your Zoom screen in the conference service and not in the chat function. We repeat this all the time, but it's important because we take those questions and then put them in a document that we call the triage document. That is a good thing to have for the moderators to see what questions we should ask from the audience. It's a great honor for me to be co-moderating this session with Professor Adashi. Eli has so far had at least 3 outstanding careers. If you have one outstanding career as a professional, then that's a great thing. If you have had 2, it's an achievement. But 3 careers means you are in a class by yourself. Eli is a pioneer in reproductive endocrinology. That was his first career with over 600 publications. Then he, after that, became the dean of the medical school at Brown University, and now is enjoying a 3rd career. He has reinvented himself as a policy, legal, and ethics scholar in reproductive medicine, focusing on genetic manipulation, particularly the topic of avoiding transmission of mutated mitochondria in women who are at risk of transmitting mitochondrial disease. So he's really the ideal moderator for this session. His current writing interests focus on the nexus of medicine, science, law, and ethics. Eli is probably the most awarded reproductive clinical scientist of our times. You can look at his bio on the web, and you can do the same, of course, for the 2 speakers today. Eli has authored articles also in the popular press, in the New York Times and the Washington Post. And a pleasure to have you, Eli. The virtual floor is yours.
Thank you, Jacques. And you are too kind, but I appreciate it. Parenthetically, I might add that before all of that, I had a career in show business. I need to fill you in.
Okay.
You're obviously not fully informed.
Oh, I'm so sorry. Next time.
So I will fill you in on that on another occasion.
Okay.
But in any event, thank you, Jacques, and I3. And perhaps a word or 2 about why we are focusing on the subject matter of today. And this, in a way, follows an earlier session where we dedicated the entire time to mitochondrial replacement therapy. And it became apparent at that point that the question of PGD in particular, prior to the conduct of the procedure or part of the decision-making progress, was not fully covered, which is really why we decided to expand that part of the story and to have the 2 experts who are with us today. The first speaker, Professor Jus St, is from Adelaide University in Australia, and he will give us a short course in the reproduction and physiology of mammalian mitochondria. Jus leads a research group focusing on the understanding of how mitochondrial DNA is transmitted and replicated. The second speaker is Professor Bert Smeets, who is joining us from Maastricht University in the Netherlands. Professor Smeets is a professor in clinical genomics with focus on mitochondrial diseases. Jacques, I think it's probably time for you to introduce our first speaker.
Thank you, Ali. The first speaker is Jus St. And he's talking to us from Tasmania. But he leads the mitochondrial genetics group at the Robertson Research Institute at the University of Adelaide, which is a few hours flying away, I think.
Yes.
His research focuses on understanding how mitochondrial DNA is transmitted and replicated. He uses a variety of assisted reproductive technologies and embryonic stem cell models. He has described mitochondrial DNA replication events in oocytes and embryos and undifferentiated and differentiating embryonic stem cells, and shown why they are important developmental outcome. And he will talk about this and other aspects of mitochondrial reproduction today. He's currently developing pig models of autologous mitochondrial DNA supplementation to enhance developmental outcomes and reproductive strategies to prevent the transmission of mutant mitochondrial DNA from one generation to the next. It really is a real pleasure to have you here, Jus. I know it's early in the morning, just past 6 a.m. in the morning, and you will talk about the role of mitochondria, mitochondrial DNA in reproduction.
So thank you very much indeed, Jacques, and thank you to both you and Eli for inviting me to come and talk to you today. So I'd like to start by showing you that mitochondria come in very different shapes, numbers, locations, and sizes within cells. Here you can see a mature skin cell. The nucleus is stained in blue, but the mitochondria is stained in green. And you can see here that these mitochondria don't have distinct profiles of their own, but they interconnect and fuse and form complex networks. And this is typical of a cell which would use their mitochondria to generate a large amount of their ATP. However, a sperm cell, for example, which you're all very familiar with, will have very few mitochondria present, and the mitochondria are all isolated in the midpiece of the cell. On the other hand, the oocyte has very distinct round type mitochondria which don't interconnect or fuse. And as you can see from this image here, the mitochondria are either round, oval, or very slightly oblong, but they're not dense, they're not dark in nature, which suggests that they don't have, they're not capable of generating large amounts of ATP, and that their ATP of oocytes might come from other places. Now, one can fertilize an oocyte, as you well know, and generate a blastocyst. And then from the inner cell mass, one can harvest the inner cell mass and generate human embryonic stem cells, for example. And here you can see the mitochondria present within a human embryonic stem cell. And they're all isolated to one end of the mitochondria in their undifferentiated state. Some of these mitochondria have maintained their sort of round or oval-like structures, and some are moving slightly more in favor of the elongated mature type mitochondria. But as these human embryonic stem cells go through the process of differentiation, the mitochondria start to migrate from one pole to the other and surround the nucleus. So as they progress to a fully differentiated state, as you can see here for a beating cardiomyocyte, which was found on the culture dish, the mitochondria pack out the cell quite considerably. And you can see here regions of very active mitochondria, which staying more lightly, and more inactive mitochondria, as you can see here, they're staining in a dark manner. And this suggests to us, and I think there's a substantial amount of evidence to support this now, that not all the mitochondria within a mature cell would be producing ATP all the time. And in fact, there's sort of like a rotational basis, or there's selection for specific regions or mitochondria from certain areas within the cell to produce ATP. They shut down and they move on to, and their colleagues come in and help. So here's a cartoon, if you like, of the mature mammalian mitochondrion. It has an outer membrane, and then inside there's the inner membrane. And depending on the needs to generate ATP through the mitochondrion, this membrane, inner membrane, will fold. And if it folds in very densely, it will produce cristae, which suggests that the mitochondrion are generating lots of ATP for the cell in which they inhabit. But these folds in the wall then result in there being a matrix. And the matrix houses the energy generating pathways, such as the electron transfer chain, but also housed within the matrix are the mitochondrial ribosomes and copies of the mitochondrial genome. And these can range from anything from 1 to 10 copies being present, depending on the cell type. But the interesting point about one of the key energy generating pathways being on the inner side of the inner membrane is that, for example, the electron transfer chain can pass electrons into the intermitochondrial membrane space and set up what we call the mitochondrial membrane potential. And if you like, that's really the electric fence around the mitochondrion to ensure that it maintains its integrity. So we require large amounts of ATP every day for our cells and our tissues and organs and our whole bodies to function normally. And we produce around 100 kilocalories of energy an hour, which is similar to what a light bulb might produce. So we really are switching something on here. But to do this, we require 380 liters of oxygen to be consumed. And in total, we generate, in a normal average state, this is not for a high performance athlete, for example, around 65 kilograms of ATP per day. And if we were to go to Sigma and buy this, it would cost us $3 million a day. So it's worthwhile in maintaining our mitochondria in very good orders. So the mature mammalian mitochondrion forms highly structured networks, but they have multiple other roles as well. They act as calcium stores and regulators. They initiate key processes such as steroidogenesis by breaking down cholesterol. They provide balanced free radical activity, so they generate free radicals which may be used as second messengers, but there are also enzymes which control the levels of these free radicals. They're important regulators and mediators of key cellular functions such as apoptosis and necrosis. And they now today thought to be major regulators of the epigenome. So, for example, byproducts of some of the energy generating pathways act as cofactors in modulating the transition from DNA methylation to DNA demethylation. And they're also involved in as regulators in other epigenetic functions. They mediate innate immune responses. And as I showed you a minute ago, mature mitochondria have multiple copies of the mitochondrial genome per mitochondria. And this is important, right? That there is a large amount of mitochondrial DNA in some of our cells. So cells have multiple pathways for generating energy. One of the key pathways is glycolysis, where an initial investment of 2 moles of glucose is broken down to produce 4 molecules of ATP. And this is the energy generating pathway that's often favored by fast replicating cells, such as embryonic cells, embryonic stem cells, tumor initiating cells, and blood cells. But the byproducts of glycolysis can also pass into the mitochondrion, and they can be broken down further by other pathways, such as beta oxidation and the citric acid cycle. And they generate electron donors that pass into the electron transfer chain, which is the cell's major generator of ATP through the biochemical process of oxidative phosphorylation, or OXPHOS. And of the 2 moles of glucose that was initially invested into this process, 34 molecules of ATP are produced. And this is the pathway of energy production that's favored by cells that have complex functions, such as heart, muscle, and brain cells. They may not need energy very quickly, but they certainly need large amounts of energy, and that's why this process is more effective for them. But as I mentioned to you, oocyte mitochondria are very distinct. They do not form networks, and they are not fused. They're single round oval-like entities, and they produce low levels of ATP through OXPHOS during oocyte maturation. But they tend to produce higher levels of ATP through OXPHOS at later stages to support processes such as meiosis, the first and second meiotic divisions, the extrusion of the polar body as the egg readies itself for fertilization. And they also contribute to the fertilization process by regulating calcium oscillations, and they produce quite high levels of ATP throughout pre-implantation development up to compaction. Once compaction is reached, then there's more of a switch to glycolysis by the developing embryo. But interestingly, oocytes have high numbers of mitochondria, but they only possess 1 to 2 copies of the mitochondrial genome, which really gives them a very distinct profile. And it's thought now that the mitochondrion itself might be the vehicle for which the maternally only inherited mitochondrial genome can be transmitted through from one generation to the next. Because if the mitochondrial genome were to lay bare within the cytoplasm of the egg, it would be destroyed by enzymes. So the mitochondrion acts as a protection mechanism and also a vehicle for their transmission. So the human mitochondrial genome is around 16.6 kb in size, and it's double-stranded. It encodes 13 of the subunits of the electron transfer chain, and interspersed between each of the coding genes are tRNAs, of which there are 22 in total. The genome also encodes 2 rRNAs and has one major non-coding region, which is called the D loop or the displacement loop. And this is the site of interaction for the nuclear-encoded transcription and replication factors that translocate to the mitochondrion to drive first transcription and then replication. The D loop also has 2 hypervariable regions, which provide us with our unique genetic maternal identity. So they define our maternal ancestors and the maternal lineage that we belong to in terms of our mitochondrial DNA content. So as I mentioned, the mitochondrial genome encodes 13 of the subunits of the electron transfer chain. And indeed, each of the subunits, except for complex 2, has 1 or more of its genes encoded by the mitochondrial genome. But importantly, we should also remember that the electron transfer chain is the only piece of apparatus within our cells that's encoded by 2 genomes, because the majority of subunits of the electron transfer chain are encoded by the nucleus. So we also know that the mitochondrial genome is highly susceptible to mutation and to deletion. Professor Smeets will talk to us later about some of the pathogenic mutations that are inherited that lead to mitochondrial DNA disease. But we also know that there are a number of spontaneous or de novo, or sometimes called somatic mutations and deletions that exists within cells and also within oocytes and embryos. And some of the early work that was produced, and some of it came from Jacques Cohen's laboratory and also from others, showed that there was a single deletion, which was 4977 base pairs in size, commonly known as the common deletion, that was prevalent in human oocytes and embryos. And there was a suggestion that this large-scale deletion was more likely to be found in oocytes than embryos. Others found an association with the prevalence being higher in oocytes from older women than younger women. But what was also interesting was that it was found later on that there wasn't just the common deletion present, but there were multiple mitochondrial DNA rearrangements that could be detected in oocytes. And indeed, there seemed to be a prevalence of more of these multiple mitochondrial DNA rearrangements in the oocytes than were found in the embryo per se. Other studies showed that there was a single base pair mutation which seemed to be present at very low levels in oocytes of younger women, but significantly increased in presence in women of older reproductive age. But another publication which highlighted something very interesting was that the mitochondrial mutations are common in oocytes, and they can range from anything from less than 5% to 50% prevalence or presence. So in other words, it could be that the oocytes of some women will have up to 50% of their mitochondrial DNA molecules harboring a mutation of some sort or another. So we've been very interested in how these various different rearrangements might impact on oocyte quality and embryo development, and whether they would be transmitted through to the offspring. And because our studies involve transgenerational analysis, it's very hard to conduct this in the humans. So we've chosen to use the pig and the mini pig to conduct these studies. And one of the reasons for choosing this particular model is that its organs, physiological and pathological systems are similar to the human. Its germ cell and DNA methylation patterns are very similar, as are its embryology and development. It produces large litters, which allows fertility to be monitored, because litter size is an index of fertility, but it also allows us to use several animals that all come from the same endometrial environment, but maybe have different mitochondrial backgrounds, for example. But we also know that this model has longer periods of gestation, which means that we can be more exact in our intervention during early development. But importantly, from our perspective, the mitochondrial DNA replication and reduction events that we observe in human oocytes and embryos are very similar in the pig. So from our perspective, it seems to be a good model to use. So we developed a mini pig colony, and we sequenced out the mitochondrial genomes, and all of the members of that colony came from one female founder. So we're analyzing what happens on a multi-generational basis through one maternal lineage. And we've identified 4 single base pair deletions within this model. And what was interesting was that for these 2 single base pair deletions here, the ovary and the granulosa cells possess similar levels to immature oocytes, maturing oocytes, and pre-implantation embryos. But when we looked at the presence of these other 2 deletions, we saw that there was not a substantial increase in the levels of these deletions, single base pair deletions, in the ovary and the granulosa cells, but there was a larger level of these there was a higher level of this deletion present in maturing oocytes and pre-implantation embryos, and for this deletion here in immature oocytes and maturing oocytes, but not in pre-implantation embryos. So it seems to be that these variants that we might be looking at could be present at different, or they could be manifestly present at different stages during development. So we took these deletions and we looked across the animals we had in our colony, and we looked in a large number of tissues and cell types, because we know that mitochondrial DNA does not segregate neutrally. And what we found was that for each of the deletions, across a large number of animals, that they never went above a certain level. So for this single base pair deletion here, it was around 16%. Here it was 9%, and here these were under 5%. So it seems to be a leveling out in somatic tissues for these specific mutations or deletions that are present. But when we looked transgenerationally, we also saw the same effect. That data is published and that's easily accessible. So the question that needs to be then answered is, are the mutations and the deletions that we see in oocytes and pre-implantation embryos being suppressed if these embryos implant and go on and give rise to an offspring, or are these the oocytes and embryos that are selected against? And because of the invasive processes that we need to conduct to analyze what's happening in oocytes and embryos, these are quite difficult studies to conduct, and we're trying to find strategies to carry that out. What I also need to tell you is that mitochondrial DNA copy number is very strictly regulated during development. We know that the primordial germ cells possess around 200 copies of the mitochondrial genome, and mitochondrial DNA copy number increases exponentially during the later stages of oocyte maturation. And it's now thought that a threshold of mitochondrial DNA copy number needs to be crossed in order that the oocyte might be ready and ready for fertilization and have the potential to undergo fertilization. So the mature metaphase 2 oocyte will have around 200,000 copies of the mitochondrial genome. And we regard this as a significant investment in future developmental outcome, because we know that during pre-implantation development, there is no replication of the normally no replication of the mitochondrial genome. And others have shown that actually there appears to be extrusion of mitochondrial DNA into the embryo's neighboring environment. Now, whether that's to aid implantation or not, or whether this is just a reduction process, that needs to be fully understood. So there's no replication that takes place until the blastocyst stage. And the replication that does take place is restricted to the trophectodermal cells. The cells of the inner cell mass, which will give rise to the embryo proper and the fetus, continue to dilute out their copies of the mitochondrial genome. And this continues until they reach very, very low levels. We define it as the mitochondrial DNA set point, but these are the founder copies really of the mitochondrial genome that will be then replicated in a cell and a tissue specific manner to support a cell's specific requirement for OXPHOS later on when it's present within a mature individual. So for example, heart cells, because they have a high requirement for ATP through OXPHOS, they will have high levels of mitochondrial DNA copy, while blood cells, which are fast replicating cells and primarily rely on glycolysis, they will acquire lower levels of mitochondrial DNA copy number. But this active reduction that takes place then ensures that the primordial germ cells for the next generation receive their 200 copies of the mitochondrial genome, packaged within the mitochondria. And these are the copies which will probably be recycled through one generation to the next down the maternal lineage and are really the units of inheritance. But while all these mitochondrial DNA replication and reduction events are taking place at various different stages of development, there are also major waves of DNA demethylation that take place, de novo methylation and maintenance methylation, plus a host of other epigenetic factors that regulate cellular function. So that's got us to think that because the electron transfer chain requires two genomes to produce effective energy, there must be cooperation between the genomes for effective cellular function. And we now talk about genomic balance. We know that the nucleus has a certain amount of DNA the nucleus has a certain amount of DNA present, and this remains very constant except for as the oocyte matures to the final stages where it adopts a haploid form. But we know that the mitochondrial genome is present in different numbers in cells depending on cell type. And we know, for example, it has multiple copies and the oocyte will have 200,000 copies. And in the mature metaphase 2 oocyte will have an equal contribution in terms of DNA content from the mitochondrial genome and from the haploid nucleus. But the sperm cell has very few copies of the mitochondrial genome, only around 10. So there must be something that creates a balance. And this balance must be readjusted at various different stages during development because we see changes in maturation of the genome and the changes in gene expression that take place during, for example, oogenesis as developmental competence is established. We certainly see different patterns of gene expression, mitochondrial DNA content as pluripotency is established, and then as differentiation takes place, and then during organogenesis and also tumorigenesis as well. So we know that there are a number of factors that act on both genomes. We know that, for example, the nuclear genome is epigenetically regulated through DNA methylation, which controls the expression of genes and gives the cell a specific phenotype. We know that the nucleus controls the mitochondrial replication factors, which control the amount of mitochondrial DNA copy that's present within a cell. We know that mitochondrial haplotypes and mitochondrial DNA rearrangements can also affect the phenotype of the cell. But the mitochondrial genome will be involved in energy production, and we know that factors that are secreted following energy production, act as cofactors that regulate methylation, and there may be a pathway here that feeds back to control both nuclear methylation and also the putative mitochondrial DNA methylation that's been described these days. So here I'm showing you this process of interaction between the two genomes. We know that there are a large number of factors that are encoded by the nucleus that regulate mitochondrial biogenesis and proliferation. And then once these factors have established the mitochondrion themselves, there are a host of factors that are translocated to the mitochondria into first drive transcription and then replication of the mitochondrial genome. And we've been particularly interested in one factor, which is the mitochondrial DNA-specific polymerase and its catalytic subunit, known as POLG or POLGA, depending how you read it in the literature. And we've shown in mouse tissues, for example, and cells that the POLGA is DNA methylated in a tissue and cell-specific manner. We know that mitochondrial DNA copy number is cell and tissue specific type. And when we take these data and we see strong correlations between DNA methylation of POLG and its expression, and we see strong correlations between mitochondrial DNA copy number per cell and relative expression, suggesting that the mitochondrial DNA replication factors may be pivotal in establishing this relationship between the nucleus and the mitochondrial genome. But in terms of oocyte function and oocyte dysfunction, we've been interested in what happens when the mitochondrial genome is abnormally regulated and whether that can actually be corrected. So here I'm just showing some data of mitochondrial DNA copy number, which we did a few years ago with colleagues in Portugal, where we saw that those oocytes which had fertilized had significantly higher levels of mitochondrial DNA than those that remained unfertilized. And then when we looked at those oocytes that had rested as P2PNs and those that had fertilized as either 3PNs or 2PNs, there was again a difference in mitochondrial DNA content with those that had fertilized and those that had arrested. So we set up a model using the pig again, where we're able to, from our own cohort or from going to an abattoir, we're able to take ovaries and isolate oocytes and culture them. And we treat these oocytes with an agent known as brilliant cressyl blue, which is just a simple dye. And brilliant cressyl blue is indicative of whether an enzyme known as glucose 6-phosphate dehydrogenase, which is indicative of developmental competence, is still being expressed or not. So when an oocyte has completed its developmental growth and it's reaching developmental competence, it no longer expresses glucose 6-phosphate dehydrogenase, and consequently, the oocytes remain stained blue. But if the oocyte hasn't completed its growth and it's deemed to be developmentally incompetent, then normally it would still be able to break down the enzyme and stain negatively. And what we see is that in the BCB positive oocytes, their cytoplasmic volumes are larger, they have higher levels of mitochondrial DNA copy number, and these oocytes are more readily fertilizable. However, the negatives have smaller cytoplasmic volumes, lower mitochondrial DNA copy number, and they're less readily fertilizable. Now, some of the BCB negatives can mature to the metaphase 2 stage. And when we analyze mitochondrial DNA copy number in the positives and the negatives at the metaphase 2 stage, we see that there's significant differences in mitochondrial DNA copy number per oocyte. And this is around 100,000 copies difference between the population. We also see differential DNA methylation patterns for POLGA between the positive and the negative oocytes. But for some of you, what might be interesting is that when you stain these oocytes and you perform confocal microscopy and you look at the outcomes, you see that the negatives and the positives have probably similar numbers of mitochondria present, but the positives maintain their distinct punctate point-like, if you like, mitochondria, but the negatives don't. Their mitochondria DNA cluster much more closely together. And we think this is due to the mitochondrial DNA deficiency, that these mitochondria aren't able to form effective electron transfer chains and therefore are not able to set up the mitochondrial membrane potential to maintain mitochondrial integrity. So here you can see on this blown up figure here that you really cannot see these mitochondria in a distinct single entities. And then when you use algorithms to try and understand mitochondrial cluster shape, and you perform analysis on this, you see that there are different types of shapes to each of the mitochondria, and their location is very, very different. Again, showing that there's real differences between these two populations of eggs in terms of the perhaps function of these oocytes. You can take both populations of eggs at their metaphase 2 stage, and you can then you can look at all the genes that are expressed using the technique known as RNA sequencing. And what you see is that between the two populations of eggs, which are, and the difference between them is that one has a higher level of mitochondrial DNA copy than the other, you can see that there are 57 genes that are differentially expressed between the two populations. And the differences lead to differences in cellular and metabolic processes, which suggest then this is to do with perhaps an energy deficiency and an ability to process cellular functions effectively as a result. When you use other analytical tools, which will predict the outcome of the effects of these differences in gene expression, what you see is that in the negative, cellular development, cellular growth and proliferation and embryonic development are predicted to be hindered in the metaphase 2, in the BCB negatives, which is what one would expect to happen. Now, one can mimic this process of having too few copies of mitochondrial DNA in culture during in vitro maturation. So in this particular case, we've taken BCB positive oocytes and we've stained them with a mitochondrial specific dye, and we've used a mitochondrial DNA depletion agent known as 2,3-dihydroxycytosine, much easier to say DDC. And we can see that over 42 hours of in vitro, 44 hours of in vitro culture, there's significant losses in mitochondrial DNA copy. And what happens to the oocyte is that the integrity of the mitochondrion per se is lost, so that you do no longer see these punctate mitochondria that are present. They just all merge and mush together, a little bit like in a way that you would see in a somatic cell, but there's no active diffusion going on. But when you take these oocytes, what you find is that they either fail to fertilize or their embryos arrest during pre-implantation development, which is very similar to what happens with those patients who have oocytes that are deficient in mitochondrial DNA copy. So using our model of having BCB positive and BCB negative oocytes, we decided that we would take the BCB negative oocytes and see if we could rescue them by supplementing them with additional copies of mitochondria that were isolated and purified. So in other words, can we increase the mitochondrial investment here so that we can support the fertilization process and support the non-replication and reduction events that take place during pre-implantation development, and get to the point where there's sufficient mitochondrial DNA present to support the developing embryo until it gets to the point where mitochondrial DNA replication kicks in. So we've taken BCB negative oocytes, and we've taken BCB positive sister oocytes and isolated mitochondria from the BCB positive sister oocytes, and we've introduced those mitochondria into these BCB negative oocytes. So we thought this experiment was doomed to failure when we first started off, because when we measured the amount of mitochondrial DNA that we could actually get in through these mitochondria into the eggs, we were only able to get around 800 copies. Now, for those of you who've done a little bit of maths at school, you will know that 800 does not make up that deficiency of 100,000 that exists between the two populations. And this led us to think, well, maybe if this is going to work, this is not going to be about providing extra batteries or units of energy to support development. there must be something else that would be happening if this was to be successful. So just so you can see, this is the very small bolus of mitochondria that we can get in. This is the maximum amount of mitochondria we can get in without destroying the egg. And you can see over here that we have to zoom in to see that small bolus that's present. But the impact is quite interesting, because if you take a BCB negative oocyte and you perform IVF on it, you'll find that your blastocyst rates will be around just under 8%. But if you add this mitochondrial supplement, you get a blastocyst rate of around 31.5%. So what we've done here is we've injected in the mitochondria as we performed ICSI, and that's why we call the technique MICSI. And this is a similar outcome to if you perform the same technique on the BCB positives. But the BCB positives don't really get a developmental advantage from the supplementation process. It only seems to be the negatives that benefit from this. So it seems to be making up for some form of deficiency that takes place. So when we looked at the blastocysts and we performed cell counts on the number of cells that were present in the blastocysts generated through ICSI on BCB negative oocytes, or blastocysts generated from ICSI-derived BCB positive oocytes, and compared the cell number to those from the MICSI BCB negative blastocysts, we saw that the MICSI BCB blastocysts had a higher cell number present at the blastocyst stage. So there seemed to be a proliferative advantage to this as well. But what's happening in terms of mitochondrial DNA content in these embryos? Well, we looked at copy number right across the whole spectrum here of pre-implantation development. This is the amount of mitochondrial DNA that's present in the metaphase 2 oocyte of a BCB negative oocyte. But when we perform MICSI, what we find is that there's a significant increase in mitochondrial DNA copy number by the 2-cell stage. And in fact, this is a 4.4-fold increase in copy number. So we have, by introducing this extra mitochondrial DNA in, we have triggered a mitochondrial DNA replication event, which means that there is then sufficient mitochondria present at the blastocyst stage to support subsequent developmental events. But what's interesting is that this happens before the major wave of embryonic genome activation that takes place within the pig, and that's at the 4-cell stage. So that led us to think, well, is this impacting on nuclear gene expression in the blastocyst? And our findings suggested that that was the case, because when we performed whole genome arrays on blastocysts generated from the ICSI, BCB positive treatment, or the ICSI BCB negative treatment, and compared it to the MICSI BCB negative derived blastocysts, we saw that the MICSI BCB negative blastocysts, their gene expression profiles clustered much more closely to the ICSI BCB positive blastocysts than they did to the ICSI BCB negative blastocysts. So in other words, there seems to be a correction in gene expression as a result. And our current studies are now looking to see if we're seeing major changes in DNA methylation that take place as a result of this. We don't have the full data sets yet, so I can't really disclose that, but it's looking quite promising. So what I guess I'm just trying to point out to you in summary, and this is my last slide, is that we have a number of factors that are responsible for interaction between the two genomes. But we know if we alter the DNA methylation status of the nucleus, we can perhaps alter, and we've shown in tumor models that this is the case, that we can alter the status of the mitochondrial DNA replication factors, which then impacts on the amount of mitochondrial DNA copy that's in any cell at any given time. But in turn, increasing or decreasing mitochondrial DNA copy within a cell, whether it's a gamete or whether it's cells of the embryo, or whether it's in a mature cell, will impact on the energy production and then the metabolism of that cell. And the byproducts of metabolism can influence DNA methylation of both genomes, but specifically of a nuclear genome, which will then affect the gene expression profiles of that cell and typically the phenotype of that cell. So I'd like to stop, and I'd like to acknowledge my colleagues throughout the world who've contributed to this work, and I'm happy to take any questions. Thank you very much for listening.
Well, thank you. This was lovely and very thorough. It illustrates the incredible complexity of this organelle and what's happening below the surface. So thank you very much for this tour de force. The way it works, I will just ask a single question now. The other questions will follow during the question and answer session. Professor St. Jin, you doubtlessly noted the recent report in Nature of the development of a base editor for mitochondria, which was a novelty, and raised at least the possibility that it could serve a clinical purpose in the management of those individuals or those families wherein there is heritable mitochondrial DNA disease. However, in a subsequent editorial or in a commentary that essentially accompanied that paper, several good points were made about the limitations of our abilities to edit either the oocyte or the fertilized oocyte, let alone the various stages of the embryo as we progress towards the blastocyst. Can you comment perhaps on your outlook on this, given your expertise really in the fine points of mitochondrial biology in this area? Can you see a potential there, or do you see limitations there? Where do you come down on this issue, which in theory would compete with replacement therapy?
Yes, so I think currently that technology is limited by its ability to deal with large numbers of genomes that would be present. So if you have, for example, an oocyte or an embryo that had, say, around 50% of its mitochondrial DNA molecules that carried the mutation, that's a large amount of work for one of these technologies to perform. And that's so if you have 200,000 copies of the mitochondrial genome, you have to edit 100,000 of those copies if there's a 50% mutant load in there. So that's a large amount of work to be done in a very short period of time. Now, if you're targeting a somatic cell, which has got a mutation, for example, in the nuclear genome, the effort for the editing system is far less per cell. So I think that's where the rate limiting step with this is, but I don't think it's out of the question. I think it's just a matter of greater scientific endeavor to find a way of optimizing this system to allow that to happen. I think the biggest problem is going to come in terms of whether society is going to consider this to be morally or ethically acceptable, because for some reason there seems to be a greater hatred amongst certain sectors of society for gene editing than there would be for taking a nucleus from one egg and putting it into a donor egg. So, and I'm quite confounded by that, because you're editing a single base pair. You're in that instance are not transferring a whole genome from one egg to another. But but in that context, that's a debate that society has to have and to determine whether they. And that's really also all about educating society about what's really going on, and allowing the science fiction angst people to be tempered in their explanations that they give about what's going on.
No, thank you very much. And of course, we share your puzzlement, but that is probably a topic beyond the scope of today's discussion. Thank you very much.
Well, thank
Then
you.
I'd like to move on to the introduction of Dr. Smeets, who will speak to us about reproductive choices for mitochondrial DNA diseases. For those of you who do not know, Dr. Smeets is a professor of clinical genomics with a focus on mitochondrial diseases. He is an internationally distinguished clinical molecular geneticist. He studied molecular biology at the University of Nijmegen, where he did a PhD on myotonic dystrophy. And for 10 years, he worked in Nijmegen and since 1995 at Maastricht University, where he became professor in 2010, combining research with DNA diagnostics. His research initially focused on inherited neuromuscular and kidney diseases, but later concentrated on the genomics of mitochondrial disorders. His research involves identifying genetic defects by next-generation sequencing, studying the pathophysiology and mitochondrial DNA bottleneck mechanism in cell lines, induced pluripotent stem cells, and zebrafish models, developing new treatment options such as compounds or autologous stem cells, and preventing the transmission of mitochondrial DNA diseases either by prenatal diagnosis, pre-implantation genetic diagnosis, or whole exome preconception screening. His research is funded by the national and international funding agencies, and he published to date over 200 original research articles, reviews, and book chapters. So, Reproductive Choices for Mitochondrial DNA Diseases by Professor Smith. Thank you very much for joining us today. Please proceed.
Thank you, Professor Adashi. Thank you for the extensive introduction. Indeed, I will continue now on the issue of mitochondrial DNA diseases and the way they are transmitted. And a large part of my presentation you can find back in a thesis, which is shown on the left-hand side of my slide. It's from Suzanne Salleveld. You can download this thesis. You can also ask it from her. She has her birthday today. So if you ask her today, you have to congratulate her. But she's currently in Australia, so basically she had her birthday yesterday, as I understand correctly from Jus. But she's a nice bridge between the two talks we have. So I'll first will continue a bit, giving a bit of background on mitochondrial disease and genetics, and I will not duplicate what Jus already said, so I will move a bit faster on that. And then I will go back into the more clinically related part of my talk. So, sorry. Mitochondrial diseases, they have many phases. You have children which are affected, you have adults which are affected. They can have specific symptoms, only, for example, the ptosis affecting the eyes. They can be wheelchair bound, it can be exercise intolerance. So there's a myriad of symptoms which can be associated with mitochondrial diseases. And one of the key things is that there is still not really a cure. So our research is both in looking for a cure, but also in prevention mitochondrial disease from occurring in families where we know that a mitochondrial disease is happening. And as just already mentioned a moment ago, you can have genes in the nucleus which have a role in mitochondria, and you can have genes in the mitochondrial DNA. So you can have a different genetic origin. I'll come back to that in a moment. So here you again see the whole system, the human system, which can be infected by mitochondrial deficiencies. And as just already said, the brain, the heart, the muscles. So there's a number of organ systems and tissues which can be affected. And these can be very specific syndromes. So you can have very specific syndromic manifestations of mitochondrial diseases, but they can also be much more general, like hearing loss, cardiomyopathy, diabetes. So you can have all these different manifestations which either directly point to mitochondria or which can have different causes as well. You can have multiple organ systems affected, so it can be multisystemic, but it can also remain to a single organ, like in Leber's hereditary optic neuropathy, which is a form of blindness, where a mitochondrial defect, despite being present in all your cells, only manifests in the eye. And the adagium is that any tissue, any system at any age can occur. The incidence of mitochondrial disease is about 1 in 5,000 and 1 in 10,000. As just already explained, you have your mitochondria, you have the mitochondrial DNA, which encodes part of the respiratory chain, and most of the subunits have mitochondrial DNA coded proteins except complex 2, but the vast majority of genes which are providing proteins which have an essential function in the mitochondria, they are encoded in the nucleus. And these are complex subunits. It's the replication machinery, what has been shown before. And knowing where the genetic cause is important for the way these diseases are transmitted. So if the gene defect of the mitochondrial disease is in the nucleus, then it's normally a Mendelian inheritance. So you can have autosomal recessive, autosomal dominant, or X-linked disease segregation. But if they are located in the mitochondrial DNA, the genetic defects, then you have a strict maternal inheritance. As Josh already mentioned, the mitochondria contain 37 genes, the mitochondrial DNA, and these are the genes which can be affected in mitochondrial DNA diseases. And the remaining part of my lecture will be about mitochondrial DNA diseases, because I think, well, these are the special type of disorders with respect to the way they are being transmitted, and that is quite different from the way nuclear gene defects are being transmitted. And to understand the mechanism of mitochondrial DNA diseases, you have to understand, and what was already mentioned before, that you have multiple copies of mitochondria in a cell or in a tissue. It's not like in nuclear genes that you have 2 copies and you can be heterozygote or homozygote. Here, the term is heteroplasmy. So you have a substantial amount of mitochondria and mitochondrial DNA copies, and you only are going to display disease symptoms if this threshold is passed. So if you have a certain amount of mutated mitochondrial DNA, which lead to a deficiency in your ATP production and your energy production, and then leads to disease. So you see, it's a gradual thing. So you can have normal mitochondrial DNA, which is homoplasmy, then you don't have any disease at all. You can have only a few mitochondrial DNA copies affected, which don't affect your system. But if you surpass this threshold, then disease manifestations start to occur. What you should be aware of is that these thresholds can vary among tissues. So it's not always a 70%, and it's not always for every mutation 70%. The thresholds varies among tissues and with mutations. The mutation percentage can change in time, so that's also quite critical for diagnosing these patients. If you, for example, test for mitochondrial DNA mutations in blood, if people get older, these mutations can disappear from blood, whereas they are still present in other tissues. So also from that point of view, it's good to be careful in your analysis and test tissues where you can expect the mutation to be present still. And what's what you also should realize, that the relation between mutation percentage and clinical symptoms is often not straightforward, which means that it's very difficult to predict the outcome in a patient when you want to base that on a mutation load. And obviously that's also a bit of a hurdle for prenatal testing, where indeed if you test prenatally, you define a certain mutation load and you want to predict the severity of the outcome. Well, this cartoon has been shown to you by just a moment ago. So I show it again because it's just indeed the bottleneck mechanism, which it's called. It's a bit, the bottleneck is basically here and here. So, but indeed you have an amplification of your mitochondrial DNA, and then after fertilization, you get a strong reduction again, and then an amplification again. And this mechanism, which was well explained a moment ago, is very important in the way mitochondrial DNA mutations can manifest. And that is what I will come back to in a moment. So if you have a patient with a mitochondrial DNA disease and you want to prevent the transmission of that in the next child or in the future offspring, then there's a number of ways you can try to achieve this. And I will focus in the remainder of my presentation on this. So on the right-hand side of my slide, you see basically the three options we have: prenatal diagnosis, pre-implantation genetic testing, and mitochondrial replacement therapy. And each of these possibilities have their own application in certain situations of mitochondrial DNA disease, and I will illustrate that during my talk. So I will first start with de novo mitochondrial DNA mutations. As just already explained, de novo mutations are quite frequent. And you should know that in mitochondrial DNA disease, if you have a patient with a mitochondrial DNA mutation, then in 20% of the cases, that is a de novo case. So, and that means that the mother has normal mitochondria and normal mitochondrial DNA, but she gave birth to a child with a very high, often mutation load, without being a carrier of that mutation, which means that the mutation has occurred somewhere in the bottleneck, and that is where we think that's the most sensitive period. So if you have the reduction in the germ cells, then if a mutation occurs, then the likelihood that it can reach significant numbers is higher. Then you already have the mature oocyte with 200,000 copies. Then it's less likely that you will see it. So the de novo mutations, what that means is indeed that because the mother doesn't have the mutation, that would imply that the recurrence risk is low. And that is what we see. So this is work again from Susanne Salleveld. Here you see 4 families with different mitochondrial DNA mutations. So the M means mitochondria. The number is the position in the mitochondrial DNA, and this is the base change. And we have 4 families with children who died from mitochondrial DNA disease with very high mutation load, so above 90%, 95%. Whereas if we tested the mother and we tested different tissue from her, so blood, hair, muscle, or urine, so it's the U. If you test different tissues, you don't see the mutation at all. So it's very difficult to fully exclude that she doesn't have the mutation in one of her tissues, but we feel if we tested all these different types of samples, then it's very likely that she's not a carrier and that the mutation occurred de novo in the child. And this is a bit counterintuitive because the child has such a high mutation load that it's often counseled incorrectly, and that is what I'll come back to in a moment. We did a number of analysis in their future when these parents came for children. We did some prenatal testing, and in every case we saw that in prenatal analysis that the chorionic villi or the amniocentesis were normal. So that means that the child was healthy. So Susanne Salt looked a bit in more depth to that. So we feel that the recurrence risk in case of de novo mutations is low. De novo mutations is then defined as being absent in different tissues of the patient's mother. And what we saw is that these families are often counseled incorrectly because they are counseled based on the mutation load of the patient, and then a high recurrence risk is given, whereas they should be counseled on the absence of the mutation in the mother. And the chances of having another child without the mutation is high, though, as always, some germline mosaicism can exist. And that is why we do prenatal diagnosis for confirmation or reassurance. As I said, de novo mitochondrial DNA mutations are frequent. So in about a quarter of the patients, the pathogenic mutations are de novo. That's from our own data, and that's also confirmed in literature. And what we also see in literature in the published families, that in all these cases, there is no recurrence when there is the presence of a de novo mutations in a child. we often don't have the full data because in case, we have done prenatal testing, the child is not always tested after birth because of ethical reasons. But the take-home message is, in case of de novo mutations, you can likely reassure the parents that they have a high chance of getting an unaffected second child. The situation is a bit different if you have recurrent mutations. So the mother is carrier of mitochondrial DNA mutations depicted by the red dots, and she produces oocyte, which can, due to the bottleneck, and that is can have a very high mutation load or a very low mutation load. These green ones will lead to unaffected offspring. These will lead to affected offspring. But it's a mixture, and it's fairly unpredictable what is happening. You could assume to do prenatal diagnosis in these cases, but then you enter into the problem that based on a certain mutation load, you have to predict the outcome. And then, as I said in the beginning, that's fairly difficult. So what we chose for is to do, in these cases, pre-implantation genetic testing, which would allow you to select embryos by which the mutation load is way below the threshold of expression. So you don't have to bother about the clinical systems. You know that the child will be healthy because it's not crossing the threshold, which I showed also in the beginning. Well, pre-implantation genetic testing is a way you can use for the heteroplasmic mutations. And here it's depicted how we're currently doing it. So it's the fertilized oocyte. This is the 8-cellular embryo. You pick out one blastomere and you can test whether it has the mitochondrial DNA mutation or not. Obviously, you can only do that for heteroplasmic mutations. If you have families with homoplasmic mutations, that will not work because you cannot select embryos below a threshold. You should know what's the threshold, and that can be an issue because in mitochondrial DNA diseases, you have many private mutations. You have to know it's reliable. Is the mutation load in the blastomere representative for the embryo? And also the bottom line is, does a carrier produce such embryos? Because if she doesn't produce embryos below the threshold, then it doesn't make sense to proceed. Well, first thing we looked at if we could define a threshold, because we say, well, we want to just reimplant healthy embryos, so we should have an idea of the threshold. And for some mutations, like the 3243 mutation is a very common one, you have enough family data available to define the threshold, and you can say, well, there it's 15%. But for a lot of other families, you don't have sufficient data to define what's the specific threshold for that specific mutation. So what we reasoned that in any case, if there is enough healthy mitochondria, you should be able to cope with any mutation. And that is something where Debbie Hellebreakers worked on. She looked at all mutations. She took them all together. She looked at muscle data, which is the tissue where we think that the mutation load is most constant. And then she found that if you have a mutation load below 18%, then the chances of being unaffected is more than 95%, irrespective of the mutation. And this figure has now been generally used in PGT, in prenatal testing, and also nuclear transfer. So it's the bottom line if you cannot define a specific threshold for that specific mutation. Well, how does it look like? This is a woman with the 3243 mutation. She went 3 cycles. The threshold for that mutation is 15%. Every column is a single cell of an embryo. And these obviously are embryos which we dissected afterwards because they are above the threshold of expression. And what you then see that here you have 2 embryos which have a mutation load way below the threshold of expression and which can be transferred and will be giving a healthy child. In 2 cycles, the woman has these embryos. In this cycle, she doesn't. And what we were also keen on is whether the blastomere would be represented for the embryo. Here you see, for example, an outlier. If you type this one, it's much more than the other cells of the embryo. But in general, you see that the levels in single blastomeres are quite constant. Here you see another example of this. Again, these embryos, you have the threshold, you have embryos below the threshold. And here you even have a very extreme case where you have a woman with a pretty low mutation load herself, and she produces a lot of healthy embryos, but she also has a very few with a high mutation load. So that's typically for this type of mutation. So in this way, you can select the embryos. I think the bottleneck mechanism is made for PGT because it allows you to select the healthy embryos. And initially we did it on 2 embryos because we thought, well, you might end up having such a case. So here you have a blastomere below the threshold where the embryo is above the threshold to prevent that. But obviously taking 2 blastomeres has effect on the viability of the embryo, and it's a bit of a balance between safety and getting offspring. So we looked in more detail to that. So we looked at this mutation, and we looked at all carriers, all cycles, all embryos, and all the blastomeres. And if we put all the data together, then you would have only 1 blastomere which would deviate from all the others. And therefore we said, well, mutation analysis of 1 blastomere would suffice, as the increase in viability is much more profitable than the risk of having a wrong diagnosis. So the error rate is quite small, only 0.43% in the 1 blastomere protocols. And what you also can see is that, most of the families or the carriers, they produce embryos which are below the threshold of expression. What you have to be careful about is that you test for the right mutations, which seems to be a bit of an open statement, but it sometimes happened. So here you have a child who died at 3 years of age. He carried a mutation in the mitochondrial DNA, the 32438G mutation, but the levels in different tissues, blood, muscle, fibroblast, or urine, were pretty low. So we were not convinced that this could explain the phenotype. So we looked further for other causes of mitochondrial diseases, and indeed we saw that the person was also compound heterozygous for POLG mutations, and that's the enzyme which replicates the mitochondrial DNA, which was discussed by Jus in the previous. And these mutations might explain the disease much better. So then you end up with a situation that you have POLG mutations, you have the possibility of a mitochondrial DNA mutation, although we would consider it a de novo because the mother is not there. And here we did pre-implantation genetic testing on both the POLG mutations and the 3243A2G mutation. And 1 blastomere was used for the one analysis and the other blastomere for the other analysis. And the parents were getting a healthy child. Well, if you look at the experience of PGT, you can say, well, it has been shown to be a reliable method. It has been shown to give rise to healthy children. It's been used in the UK, in Paris, in Maastricht, in Ghent, in the US. So many groups have provided data, and this is only a brief overview. It's not complete. It's also not complete because many of the cases are not published anymore, but here you look at what was found in the embryo, and here you can see also what was found postpartum, and there's only 1 strange case which is still not resolved, where there's a discrepancy between what you find in the pre-genetic pre-implantation testing and in the embryo. This was on the blastocyst, so that was not on cleavage embryos, as I showed you just a moment ago. For the cleavage data, it's much more consistent, that you see if the embryo result matches the results which you see after birth. But there's also blastocyst data where you do have the same results. So there might still be an issue on the technicalities of this case. So if you look at the PGD data in Maastricht, Paris, and Newcastle, and I think Newcastle is still collecting this data to have it published, because I think it would be sense, then you see that already have considerable number of females have undergo PGT, a number of cycles, a lot of samples have been done. And yeah, as I said, the heteroplasmy is concordant for the cells within an embryo, and the range is well defined. So that means that the technique is reliable. And what you can imagine, that if the heteroplasmy in the carrier is higher, then the embryo heteroplasmy will be higher as well, which will downscale the PGT success, because you will not have sufficient embryos below the threshold to put back. So in summary, how far will pre-implantation genetic test us in mitochondrial disease? So I think carriers of all heteroplasmic mitochondrial DNA mutations have a fair chance of having healthy offspring by applying PGT. It's technically safe and reliable. We can estimate a safe cutoff mutation percentage at which the risk of being affected is acceptably low, so below the 18%. We expect that most mutation carriers will have oocytes below this threshold, and I will come back to that later. You can argue that the exact cutoff mutation percentage can be determined by case counseling, and there's also national differences between regulations in countries. You could argue by selecting male embryos that you could also eliminate mitochondrial disease in future generations, but obviously is an ethical issue. And blastocyst or trophoidect biopsies is still under discussion. There is some controversial data, but it's not validated yet. But I know that our group in Maastricht is now trying to look into that. And so far, the data seem to be more promising that you can also move there to trovectodon biopsies. Well, I only will touch briefly, as we're not doing this ourselves, the mitochondrial DNA replacement therapy, and I know that that has been discussed previously, but if you have high heteroplasmic of homoplasmic mitochondrial DNA mutations, then the woman will only produce affected oocytes, which will, after fertilization, will lead to affected embryo. So you have to exchange them. There's no other ways to get healthy children. And for exchanging that, the techniques might be familiar for you. This is if you do it in the metaphase 2 stage. Here you have a metaphase 2 oocyte with healthy mitochondria. This is the patient who wants to have a healthy child. You extract the spindle, you remove the spindle from the other oocyte, you put in the nuclear DNA of the carrier in the oocyte with the healthy mitochondria, fertilize it, and it will give rise to healthy offspring, where the mitochondrial DNA comes from the donor and the nuclear genome comes from the parents, the biological parents. In Newcastle, they do it at the pronucleus stage, but the principle is the same. So the pronucleus, you extract the pronuclei, you remove them from the healthy cell, and you put in the pronuclei from the parents with the affected mitochondria, and you get the possibility to have a healthy child. How far will MRT bring us? So these spindle pronuclei and also polar body genome transfer are capable of generating mutation-free embryos. So it's a solution for all mitochondrial DNA mutations. There is an issue of the carryover, so there is a minimal amount of carryover due to the fact that you take the pronuclea in a little bit of cytoplasmic environment. So it's unlikely to cause disease, although reports have been shown that you might have some increase in the mutant mitochondrial DNA, which leads to a switch from the healthy to the mutant one, and it's still not very clear what the mechanism is and how frequent it is. Obviously, you're manipulating with the germline, but ethically, MRT is considered acceptable. So it's been published by Breda and also by the Nuffield Council of Bioethics. As so far, the tests are promising in primates, mice, fertilized oocytes, and few patients. The method seems safe, but obviously issues remain on long-term effects, epigenetic issues, because you're taking the mitochondrial DNA away from its normal environment. You can use it for heteroplasmic and homoplasmic mutations, and in the UK, it's only allowed if PGT, if pre-implantation genetic, is not possible as an alternative. So in the UK, the rules are that it should be severe disease and PGT should not be possible, and then it's allowed to go further. But also there, different countries might have different regulations. And obviously for this, you need a sufficient donor oocyte or zygotes to be able to go that way. So as said, in counseling these patients, it's important to advise them whether PGD, whether that is a good possibility to get healthy offspring, or that the chances are so low that MRT starts becoming the preferred choice, although we still don't know how easy it is to generate offspring in that case. But for that, to predict these chances, Patrick Lindsay, our bioinformatician, he proposed a model in which you include the number of embryos that could be reimplanted, so which gives you the chance of having unaffected offspring. Is obviously related to the number of oocytes obtained per cycle, the maturation, and the fertilized oocyte percentage, and the probability of being under the 15% or 18% clinical threshold. So in this case, it was 15%, so because he modeled that for the 3243 mutation. He used the Kimura distribution, which is a statistical model of doing that, and then indeed you get these type of graphs that, based on the mitochondrial DNA heteroplasmy level of the carrier, you can calculate how many healthy oocytes such a woman produces, and you see at the moment it gets higher, the number will be lower. And what we generally reason is that PGT makes sense if at least you would have per cycle one embryo below the threshold which could be reimplanted. If that's not the case, then you might wonder whether it makes sense to go further. This Kimura distribution is based on random distribution, so it means that the mitochondrial DNA mutations distribute randomly during the bottleneck, but we know that that is not the case. So we looked also at our embryos and the mutation load in single cells of one of the embryos. So these are the embryos of one carrier. So every dot is the mutation load in one of her embryos. And then you see that, for example, for this mutation, the 3243, we never see a very high mutation load, and also just addressed it in his previous presentation that some mutation loads may not be capable with viability. But if you know that these cannot occur, then that would also affect your prediction model, and you can also have skewed distribution. This mutation we know goes either to the very high mutation load or the very low mutation load, and obviously that has impact on the way you counsel these carriers and the way you weigh their chances of getting healthy offspring. And that has also been then considered in the model. So if you have a truncation, so here the model is the truncated Kimura distribution, which means that for the previous slide, it's this situation where the distribution doesn't go to the 100%. Then you can include that in the model, and then you can also calculate, based on the distribution of your mutation which you observed in your offspring, the more precise the chances that you get unaffected offspring by using PGT. And obviously, and as we use that also in deciding, because I have to advise also the fertilization authority in the UK, the Newcastle Fertility Use, if they have PGD cases, and then we use these models to see whether PGD can be excluded as a possibility for these patients, and they should go for MRT. So that brings me to my last slide. So if you would like to go to future without mitochondrial DNA disease, there are ways to stop the transmission. And so it's by prenatal diagnosis in case of de novo mutations, which are frequent. Some recurrent mutations, if the mutation loads are very low, then you can also advise the parents to go for prenatal testing. PGD is good for the heteroplasmic mutations. Both methods are safe with a small residual risk. And more experimental options these days is mitochondrial replacement therapy. Spindle transfer was used in the US, pernuclear transfer is used in the UK, can be applied for homoplasmic and heteroplasmic mutations, so it would basically cover everything. The residual risk based on the carryover seems low, but experience is still limited, and also with the chances of becoming pregnant. So experience has to be developed, and based on that, you might see whether this could replace also the other options we currently have. But as I said, mitochondrial DNA disease occurs de novo in many cases. So it's still essential to work on therapy development, because we can still not prevent mitochondrial DNA disease from occurring. And that is something which is also part of the research we're doing. So that brings me back to the people working on it. It's people working on PGD. It's Joep Gerrits and Hans Evers who started that in Maastricht in the early days. Debbie Helkebrekers is currently very much involved in the PGT for mitochondrial DNA disorders, and Christine de Die does the counseling. So if you have interest in having these PGD done in our center, then you can contact them for further information. I want to thank you for your attention, and I give the word to the moderators back.
Thank you very much, Bert. That is a great presentation, a wonderful overview of all the great work that you and your associates have done in Maastricht. Congratulations. We have quite a lot of questions. A quick one for you, Bert. The use of mutation-specific thresholds does not take into account that the loads are determined from mature somatic tissue, rather than considering the chance that the mutation may amplify during early development,
that's true.
Which is something you address by referring to, I think it was Egley's work on stem cells. Yeah. But the genetic drift, of course, not using mutated and wild type mitochondria, the genetic drift was observed when using 2 wild type mixtures early on in the work. Yeah. The lab I associated was with cytoplasmic transfer. We found in 2 out of 7, we found that one of the populations amplified a lot after embryo transfer. And recently, a few months ago, I think it was in December, Pavlo Mazur from the Ukraine presented his work of pronuclear transplantation for infertility and showed that, well, first of all, they showed that the carryover was very low, probably lower than 2%, certainly in almost all cases lower than 1%. And yet 2 out of the 9 babies said, I think they had 10 babies, and 2 out of the 9 showed excessive drift of the maternal mitochondria at the time of birth. And so if you, is this, do you think this is, the difference here is because when you work with mutations, those mitochondria may not divide may not amplify very well? Is that maybe why in PGT you're being in an advantageous position?
Well, in PGD, it's you only have one background of the mitochondrial DNA. So it's the same mitochondrial DNA population, which either has the mutation or not, which is a different situation if you would do a pronuclear transfer or these type of things, because then you have 2 different populations. And that's also why people thought initially that they should be doing haplogroup matching, and that they would be comparable. But I think the one which in the UK went high in the switch was haplotype matched, so that could not be the case. People have argued that it might be replication difference, but I think that's a specific concern for pronuclear transfer or spindle transfer, but not for PGD, because that is the same mitochondrial DNA and it only differs at one position, which is the mutation. So you can have differences, but then they should be more related to selection against one of them. But it could not be the case that there's a replication advantage. And I agree with you, the choice of which tissue to choose to define the threshold, we chose for muscle. And that's also based on studies they did in Paris, that the muscle mutation load was fairly comparable what they saw in villi or in other tissues. But yeah, I do agree that you can always, that's a very difficult point. But in general, I think it seems to have to work quite well, this threshold, and it gives guidance to people making these choices.
Do you just very, very quickly, before we everyone joins and we will have a general Q&A, are you following up these children? Are you looking every year or every few years? I know it's involved, but are you doing that? Because I realize that 80% threshold is based on very solid literature searches and all that.
Yeah.
But you could still expect that it changes over time.
Yeah, we do follow them up. I showed you in the beginning, after birth, they are being tested. Indeed, if it's now, there's always an ethical issue, but it's accepted because you have that data and you need it to show that the method is safe, that you test them early on. And I think they are being followed up, but it's obviously voluntarily, so it depends also. And the numbers are still quite small, but I agree with you, it's very essential or crucial to keep an eye on these children. But so far they're doing well. The problem sometimes is we also get them from internationally. Yes, so you have had a family from Italy with a private mutation, an older woman who gets a child. Well, you will lose track of those. That is sometimes due to the where people live and how they can be followed.
Yeah. Well, thanks very much, Eli. I'll ask a few questions, and then please jump in, and we'll have about 15 minutes or so more. It's been a wonderful session so far. Question for Jus, from Mitch Sheewood from California, who says, fascinating lecture. Thank you very much. How might mitochondrial changes or deficiency be involved in frozen oocyte batch variation in developmental incompetence resulting in cleavage arrest or reduced slow blastocyst development? Of course, he's referring to IVF conditions. Do you think there could be a role here?
Yeah, well, I mean, that's an interesting point. So if you have oocytes which are deficient in mitochondrial DNA, that they may, or pre-implantation embryos, that if they're deficient, that their mitochondria would be affected, and therefore the freezing protocol may actually have an effect on the mitochondria per se. So when you thaw out these entities, they may be more affected than, or they may not have the protection for their mitochondria, which you might want them to have. So that's an interesting point, which actually I hadn't really thought about in great detail, but it's something that you may want to think about in terms of treatment, especially for patients who are of older reproductive age.
Yeah. And maybe what can come up, since we know that we can pick up DNA from embryos now in the blastocyst as well as in the culture medium. Maybe this can be assayed in the culture medium. Similar, a question from Pavla Pachova, who is asking you, at which point during oogenesis are these mutations occurring, you think?
Is that for me or for Bert?
Yeah, so
okay, I'll have a go and then Bert can have a go, I guess, right? So, yeah. So in the so I mean, I think that's probably the question that everybody really wants to answer, because we know that there are reduction events which take place, and we don't know if there's selection for, at any stage during development, for preferential selection for molecules that got mutations or not, or whether it's a completely random event. But perhaps it's to do with, excuse me, perhaps it's also to do with the energy status of that cell at any stage of development. So Bert showed quite nicely the changes from glycolysis to OXPHOS when mapped over the top of my mitochondrial copy number graph. And so if copy number is decreasing, say early post, so when glycolysis is switched on, when the cell's going through high levels of proliferation, it might be that those molecules with the mutation, those cells with the mutation might be preferentially selected for, because, and those molecules preferentially replicated, because that would promote the use of glycolysis, because the cell would not have the potential to go to OXPHOS. And then it's only later on that the implications for the mutation come in, because then the cell or the tissue that would be affected would not then be able to sustain high levels of OXPHOS production because of the mutation that's present. So it might be that actually this bottleneck that comes into play may be metabolically driven as well, so that you have mutations being preferentially selected for at that stage, because if you have high levels of mutation present, you have glycolysis, therefore you promote proliferation of the cell, because glycolysis is the mechanism, is the energy generating pathway that's associated with proliferation. And this is something you also see in tumorigenesis, where you have partial shutdown of OXPHOS, and you go for this Warburg effect, where you have low levels of OXPHOS present, but high levels of glycolysis, which promote cell proliferation. So we may be seeing a phenomenon very similar to what happens in a tumor at that stage. And that's why sometimes studying tumors and embryos in parallel sometimes, provide a lot of information about, especially mitochondrial dynamics and mitochondrial metabolism.
Right.
Can I comment on that? Or
Sure.
Yeah, we studied that specific question also in zebrafish. It's not something I presented, but we looked at oocytes from zebrafish. We did next generation sequencing to see what de novo mutations do occur, and we saw that they were quite random, and that they were equal to the bit of the error rate of the polymerase gamma. But with next generation sequencing, you obviously have the threshold of detection. So we said it's 1.5%. So, and what we saw is indeed that if you go above the 1.5%, that would require that your mitochondrial DNA copy number would have to go below 65 copies or something like that at the bottom of the bottleneck. And that seems to match. It's not causality, but the numbers seem to match. So the number of oocytes or primary mitochondria with would be below 65, match the number of mature oocytes where we found a de novo mutation, which was random. So we don't know yet how it will proceed. But it seems to be a mechanism where the oocyte or the primordial germs is most sensitive when, I think, no, I should rephrase it. Mitochondrial DNA mutations occur all the time, but you're only going to see them if they occur when the copy number is very low, and if what just said, they are enriched in a certain way. But they will happen all the time at every moment, but usually you don't see them, but you only pick them up when they are at a critical point during development and then enriched by amplification.
Yeah. So in zebrafish and also in the pig, you're looking at normal material. Now, we are not that lucky, and so those mutation studies in the human, there's only a few papers. That's usually spare material, so that they're abnormally they're eggs that never matured properly or didn't fertilize, or presumably didn't fertilize, or fertilized abnormally, embryos that stopped developing. And so the literature says that enormous numbers of clinical mutation rates in these early embryos, but I don't think that people have looked at normal embryos and mutation rates, or presumably normal embryos.
No.
So it's a fascinating area.
Yeah.
Ali, if you would like to take it from here.
Yes. So there's multiple questions, of course, to both speakers, but let me tackle some for Professor Smeets. Shira Artsy asks several questions. Well, one of them is, are there any known factors associated with the chance of de novo mutations? Or is this just a matter of chance?
Yeah. That is a good question, which I don't have the answer on. You cannot really tell. It's the same question again, which we try to address in zebrafish, but obviously we try to validate indeed the zebrafish model, showing that the whole mitochondrial DNA bottleneck and everything was similar to humans, but you never can be fully sure. But what we saw in zebrafish is that there's a high difference in oocyte copy number, and maybe Jus can comment on that, in an individual woman. So it was not that you would have, or an individual female fish, I should say. So it's not that you would have fish with high or low. It was a huge distribution. And if that would be the same in women, and if our hypothesis would be valid that it's critical if you have a low amount, that would mean that every woman would be at risk, and it would just be bad luck that you had a low copy number mitochondrial DNA, which would be your future child and which would be more at risk of having a de novo mutation. But that is indeed, there's a lot of speculation in this answer. But what struggles indeed was that there was that huge diversity in copy number in different oocytes of the same fish. And I think that probably, in human, obviously these data are not that extensive, but I think that variation is still there. But it's not, you might have situations where there is a familial cause for having a reduced copy numbers, but these are quite rare, I must say. Yeah.
So somewhat of a ongoing mystery, I guess,
Yeah.
is perhaps the bottom line.
Can I just interject? Sorry,
Sorry.
can I just interject very briefly? Sorry, that's a very important point that Bert raises there. You know that we talk about percentage mutant load or whatever, but actually we should also be talking about the number of mutated copies against the number of wild type copies, because as Bert rightly says, you know, if you've got an oocyte that's got 50% mutation, 50% mutation loading, but it's got 200,000 copies of the mitochondrial genome, there are in fact 100,000 copies which are wild type and can pass through. And that in itself would be a very interesting outcome. And we tend to ignore that because we get stuck on the percentage and not the actual numbers. And that's a very interesting study that could be, you know, would provide perhaps more data than we actually have, more insights than we actually have into the transmission of mitochondrial disease.
Well, thank you for highlighting that. Bert, did you want to expand on that?
No, it is again in the fish we see indeed, as just also mentioned, a limitation in the copy number in the oocytes, and below that copy number you don't see them anymore. So there's obviously, and then we saw, well, you need at least 50,000 healthy mitochondria to be able to get appropriate embryonic development. That is, and I think in this way you can see what is the energy amount you need, and if you have a mutation, obviously you need more mitochondria, which can compensate for the deficiency. But that gives you a bit of a feeling what is needed for normal development, which for the work of Jus might be important also, that might also give an indication what you need as energy reservoir for proper embryonic development, irrespective of mitochondrial DNA diseases. It gives you a clue about what you need, what a healthy cell would need to develop normally.
Just maybe for the benefit of the audience, Bert, can you explain why you choose zebrafish for these type of studies?
Yeah, we chose zebrafish because they have huge numbers of offspring. They are cheap, and we can isolate the different stages of germ cells. So if we let them express certain genes during development, we can isolate the primordial germ cells. We can isolate all stages of maturity in the oocyte. So you basically can fill in the bottleneck scheme which just showed, and we showed that the bottleneck and the lowest level, it's very conserved during evolution. So the amount of mitochondrial DNA at the lowest point of the bottleneck is conserved through crickets, through fish, and even though with the fish you start with millions of mitochondria, so the amount is much higher in those cells than in human oocytes. But we think because of the ex vivo development, so they don't implant, so they need more to be able to survive. But mechanistically, it seems very simple. Obviously, we cannot test implantation, but we do want to see how they develop. And I think it's a very nice model, and it's easy to manipulate. We were able to reduce the copy number by adding morpholinus against TFAM, so it's one of the proteins just mentioned. And then you can reduce the copy number and see how that impacts development, but also how that impacts the further transmission. And the next thing what we would like to do, and that's what you were alluding to in the beginning, is to create mutations in that mitochondrial DNA. But also that seems more practical to do, but at least it would give you the numbers to draw reliable conclusions, because what you see these days, that a lot is based on a single mouse model, on a single mutation, and these conclusions are amplified, but yeah, you still don't know how generally applicable these things are, because you don't, in these models, you cannot have the figures you could have in zebrafish. But that is the reason why we moved to zebrafish, to have a cheap, quick model, easy to manipulate, and which has a bottleneck mechanism which is very much conserved and comparable to humans.
Great. Well, thank you for that explanation.
Really, every model has its upsides, although one cannot overemphasize the similarities between the porcine model and the human model, which is obviously a terrific advantage.
Sure.
Same time, of course, it's quite obvious that it's far more complicated to work with that model compared with zebrafish that swim in your laboratory aquarium.
Yeah, that's true.
But such is life, and I think both of you are doing important work, and both models obviously answer important questions. Jacques, you wanted to intervene?
Well, so yeah, so maybe just one question for each of you, and then we wrap it up. We have a lot of questions here, and I'm sorry to the audience that we didn't get to all of it. There's a question here for Jus by Jeremy Cheng from Atlanta, who's asking, what's your opinion about the mitochondria DNA copy number, since you've talked about it as a predictor of embryo viability in the pre-implantation stage embryo? You think that is a viability factor?
Yeah, so I do think that's actually there's something that might be worth assessing. And that could be done by looking at the culture media to see. I think so. What I'm trying to say is, I think it's important to see whether this active extrusion of mitochondrial DNA and this reduction in mitochondrial DNA copy number takes place, because I think that's a important developmental mechanism. And if there is premature replication of the mitochondrial genome, for example, during pre-implantation development, it would suggest that the interactions between the nucleus and the mitochondrial genome are not running efficiently. So I think that in some of the studies we've done, for example, we've looked at individual blastomeres in the porcine embryo and seen that there are reductions that take place, and that these perhaps are indicative of embryos that will progress through to blastocysts. But if you can see this in the culture media, then I think that's a good indicator that your embryos will develop normally. So,
right. Maybe Ellie, you see a good question for.
Yes, there are many good questions. So again, apologies to the audience, but another question to Professor Smeets from Cecilia Fernandez, who asks, what methodology is used to quantify the load of mutation in embryos? Is this real-time PCR? Of course, you addressed that already, but perhaps a clarification may be helpful here.
Yeah, yeah, you do that single cells, although it's not really critical single cell technology because you have that many copies of the mitochondrial DNA. We did it by PCR and cleavage, but it's a mutation-specific assay, which, yeah, you can optimize any kind of mutation-specific assay you like. And you just have to be sure that you have a proper control, and then it's a cleavage, and then you measure the amount. But you could also do it by NGS or whatever, what technology is available in your laboratory. And as said, because you have about 50,000 copies in such a blastome, or 25,000 copies in such a blastome, it's more easy than looking at the single nuclear genes as you generally do in PGT. So it's more convenient. Yeah. But we can share protocols if people are interested. Yeah.
All right. Well, I think coming to an end, and like to wrap it up here. Ellie, if you start off, and then I'll announce the next session.
Thank you, really, everyone, for taking the time to participate in this session and discussion. I particularly would like to thank the I3 speakers, Professor Jus St and Bert Smeets, and my co-host, of course, Jacques Cohen.
Pleasure.
While we're at it, we also want to thank the other organizing committee of this initiative. I'll mention Peter Nagy, Thomas Elliott, Shasta Sadruddin, I hope I'm not mispronouncing this, Giles Palmer, Marianne Cevetez, and today's team, Q&A triage team, which we already mentioned to some degree. So at that point, perhaps it would be time to punt the microphone back to Jacques, who will, I think, educate us on what's coming up.
Thank you very, very much, Ellie, and thank you for being an outstanding co-moderator. This session and presentations will, of course, be available on the website. That usually takes a day or so. So also, if you have a chance, don't forget to rate our lectures. A certificate of attendance will be sent to you before the next session. Next session is next week. Same time for us here on the Eastern time zone, and same time for everybody else in the United States. But however, please watch your time changes everywhere, because, you know, times are changing in Europe, for instance, and not everywhere in Europe, maybe. So please check your clocks. The next session will be about surgically removed spermatozoa. Our audience loves spermatozoa, so we'll know that will be a high attendance. And we have two outstanding speakers, Sherman Silber and Audie Bray. It's all on the website, check it out. And there will also be a panelist, Professor Alan Goossens from Belgium. And the moderators will be Peter Nage and Giles Palmer. And the session is called Discovery of Sperm. So with having said all that, thank you again, speakers. Thank you, Eli. And see you again soon next week. And all of you be well and stay safe. Thank you.
All right. Thank you.